Welcome, everyone. Today we are exploring comparative genomics, a field that helps us understand life by comparing genomes across species. Every organism carries a genomic blueprint, but those blueprints are not completely independent. Many parts are shared, some parts are modified, and some parts are unique. Comparative genomics studies these similarities and differences. By comparing genomes directly, scientists can identify regions that have remained conserved over long evolutionary time and regions that have changed more rapidly. Conserved regions often point to essential biological functions, while divergent regions may reflect adaptation, specialization, or evolutionary innovation. If you look at the visual on the slide, the key idea is that genomes can be compared like biological manuscripts to reveal shared ancestry and evolutionary change. So the central question becomes, why is it so useful to compare the genome of one species with another? Comparing genomes is powerful because evolution leaves clues inside DNA. Some genes remain highly conserved across species because they perform functions that are essential for life. If a gene has remained similar in humans, mice, zebrafish, and flies, that usually tells us that the gene is biologically important. Comparative genomics also helps us predict gene function. For example, if a gene in humans is poorly understood, but a similar gene in another organism has already been studied in detail, that information can guide our interpretation. This is especially important in model organism research. Knowledge gained from mouse, fly, or yeast genetics often helps us understand human biology. If you look at the message on the slide, comparative genomics is essentially helping us read the evolutionary footprints written into genomes. But before using databases and genome browsers, we need to understand the key biological relationships being compared. There are several major ways to perform comparative genomic analysis. One is whole genome comparison, where large genomic regions or entire genomes are aligned to identify shared and unique sequences. Another very important concept is the distinction between orthologs and paralogs. Orthologs are genes in different species that originated from a common ancestral gene through speciation. These genes often retain similar biological functions. Paralogs, in contrast, arise through gene duplication within a genome. After duplication, one copy may retain the original function, while the other evolves a new or modified role. This distinction matters a lot because orthologs are usually more informative for predicting conserved function across species. If you look at the diagram on the slide, the separation between speciation and duplication is one of the most important ideas in comparative genomics. To explore these relationships visually, researchers rely heavily on genome browsers. Raw genome sequence data is enormous and not easy to interpret directly. A file containing billions of nucleotides is not something the human eye can easily analyze. Genome browsers solve this problem by converting genomic information into an interactive visual format. They allow researchers to see genes, exons, introns, regulatory regions, variants, and conservation patterns in one integrated view. This is extremely helpful because comparative genomics is not only about sequence alignment. It is also about viewing how functional features are arranged and conserved across species. If you look at the visual on the slide, think of genome browsers as navigation systems for genomic landscapes. One of the most widely used platforms in this area is Ensembl. The Ensembl genome browser is one of the most important resources in comparative genomics. It provides a highly curated platform for exploring genomes, especially vertebrates, and major model organisms. Ensembl allows researchers to examine gene structure in detail, including exons, introns, alternative transcripts, and regulatory annotations. One of its major strengths is that it integrates comparative genomics directly into the browsing experience. That means researchers can look at a gene and immediately connect it with homologous genes, conservation patterns, and evolutionary information. If you look at the slide, Ensembl is acting as both a genome browser and a comparative analysis platform. But Ensembl does more than just show genes. It also reconstructs their evolutionary relationships. Ensembl includes a set of comparative genomics tools that help researchers trace gene evolution computationally. One major feature is gene tree reconstruction. These gene trees help identify which genes are orthologs and which are paralogs. This is important because it allows scientists to distinguish conserved relationships from duplication-based diversification. Ensembl also provides conservation scores. A highly conserved sequence usually suggests functional importance because evolution has preserved it across species. These conservation tracks can highlight coding regions, regulatory elements, and other important genomic features. If you look at the slide, the key message is that comparative genomics is not only about seeing similar sequences, it is about inferring evolutionary history from them. While Ensembl is especially strong for vertebrates, comparative genomics extends far beyond animals. Biology is much broader than vertebrates. Comparative genomics also needs to include plants, fungi, bacteria, and protists. That is where Ensembl genomes becomes important. It expands the comparative framework to non-vertebrate organisms and allows researchers to analyze genomic relationships across a much wider range of life. This is useful for studying plant evolution, microbial diversity, fungal genomics, and many other biological questions. It also supports cross-clade comparisons, which can reveal how ancient biological mechanisms are shared across very distant organisms. If you look at the slide, the important idea is that comparative genomics is not restricted to closely related species. It can reveal deep evolutionary continuity across life. Another powerful browser used in this field is the UCSC Genome browser. The UCSC Genome browser is another widely used platform for comparative and functional genomics. One of its main strengths is the use of stackable tracks. These tracks allow researchers to place different kinds of information on top of each other. For example, you can view gene annotations, conservation scores, variation data, regulatory features, and comparative alignments all in the same genomic region. This layered structure makes it easier to interpret how genomic features overlap. The UCSC browser is especially useful when examining how a region is conserved across many species at once. If you look at the slide visual, the browser becomes a comparative landscape, where many data types can be explored together. Behind such browsers, however, there must also be a large infrastructure of stored genome assemblies. The NCBI Genomes database is one of the major resources for genome assemblies from across the tree of life. It includes both complete and draft genomes and covers a very large number of organisms. This provides the foundational sequence information that comparative genomics depends on. Researchers can retrieve assemblies, compare species, and use these datasets in broader evolutionary studies. The database is especially important because it acts as part of the primary infrastructure supporting genome-level research. If you look at the slide, think of NCBI Genomes as a large warehouse of genomic blueprints. Sometimes, though, general databases are not enough. Certain groups of organisms require more specialized comparative resources. Plant genomes often behave differently from many animal genomes. They can be large, repetitive, and shaped by frequent gene and even whole genome duplication events. That makes plant comparative genomics especially challenging. Phytozome was developed as a specialized platform for plant genomes. It provides plant genome assemblies, annotations, and comparative tools that are tailored for studying plant evolution, gene families, and agriculturally relevant traits. This kind of specialization is important because one-size-fits-all genomic tools do not always capture the biology of every lineage equally well. If you look at the slide, Phytozome represents how comparative genomics becomes more powerful when adapted to the biology of a specific kingdom. Now let us focus more closely on one of the most important biological concepts in this field, orthologs. Orthologs are one of the central concepts in comparative genomics. These are genes in different species that originated from a common ancestral gene through speciation. Because of that shared ancestry, orthologs often retain similar functions. This is why model organisms are so useful in biology. If a gene in mouse or zebrafish is orthologous to a human gene, studying that gene in the model organism can reveal important functional insights relevant to human biology. Orthologs are, in a way, the shared blueprints of life. If you look at the diagram on the slide, the key message is that evolutionary conservation helps us transfer knowledge across species. To identify these connections with confidence, dedicated orthology databases are used. InParanoid is an orthology database designed for detailed comparison between pairs of species. Instead of trying to analyze very large multi-species groups at once, it focuses on identifying high-confidence orthologs between two organisms. This pairwise approach is useful when a researcher wants a more focused and reliable comparison. For example, one might compare human and mouse genes, or human and zebrafish genes, to identify likely orthologous relationships. This is especially valuable in functional transfer studies, where researchers want to predict whether a gene in one species likely performs a similar role in another. If you look at the slide, the important idea is precision in one-to-one or closely related pairwise comparisons. When we scale this kind of work across many organisms, broader resources become necessary. OrthoDB is a large orthology resource that covers a broad range of life, including animals, plants, fungi, and bacteria. Its strength lies in its hierarchical organization. That means it can define orthologous groups at different evolutionary depths. For example, it can identify relationships at the vertebrate level, mammalian level, or primate level. This layered approach is useful because gene conservation can be interpreted differently depending on the evolutionary scale being studied. OrthoDB is therefore very useful for large comparative studies that span many organisms and many taxonomic groups. If you look at the slide, the key message is that orthology can be studied not only pairwise, but across structured evolutionary hierarchies. But identifying related genes is only part of the story. We also need functional context. eggNOG is a database that connects orthology with functional annotation. It groups genes into orthologous clusters, but it also provides useful functional information about those groups. That makes it especially helpful when researchers are not only asking which genes are related, but also what those genes are likely doing biologically. This is important in large-scale annotation projects, comparative genomic studies, and metagenomic analyses. If you look at the slide, eggNOG acts like a bridge between evolutionary grouping and functional interpretation. To broaden the view further, another resource that integrates homologous genes across species is HomoloGene. HomoloGene is part of the NCBI ecosystem and focuses on homologous genes across species. Importantly, it includes both orthologs and paralogs. This makes it useful for studying not only conserved gene function, but also gene family expansion and duplication-based diversification. In other words, it helps researchers understand both shared ancestry and evolutionary branching within gene families. If you look at the slide, the central idea is that comparative genomics is not only about one-to-one similarity, it is also about how gene families diversify across lineages. For researchers especially interested in animal gene family evolution, TreeFam is another key resource. TreeFam focuses on the evolutionary history of animal gene families. It provides phylogenetic trees that help researchers visualize how genes have expanded, diverged, or been lost over time. This is important because gene families are not static. Through duplication and divergence, they often acquire new members with specialized functions. TreeFam helps track these histories in a structured evolutionary framework. If you look at the slide, the phylogenetic tree is showing gene evolution not as isolated events, but as family-level biological history. This kind of evolutionary analysis is not just theoretically interesting. It also has direct relevance to medicine. Comparative genomics also contributes to clinical genetics. When researchers compare genomes and identify conserved or altered regions, that information can help connect genomic variation with disease phenotypes. This is especially useful in the study of rare genetic disorders, where identifying the affected gene can be difficult. Comparative evidence from other species can strengthen the case that a gene is biologically important and that disruption of that gene may lead to disease. If you look at the slide, the key message is that comparative genomics helps bridge basic evolutionary biology and clinical interpretation. From there, these tools support broader biological discovery. Comparative genomics drives discovery in many areas of biology. One major outcome is functional gene prediction. If a gene is conserved across species, it is often possible to infer its likely role. Another important application is identifying conserved regulatory regions. These are non-coding sequences that have remained preserved because they control essential biological processes. Comparative genomics can also support drug discovery by identifying protein regions or pathways that are essential across organisms. This can highlight vulnerable biological targets with strong functional importance. If you look at the slide, the message is that comparative genomics does not just describe evolution, it actively generates biological insight. That insight also extends into modern medicine. Comparative genomics plays an important role in modern biomedical research. It helps identify disease-associated genes by showing which sequences are highly conserved and likely functionally critical. It also helps researchers study pathogen evolution, which is very important for understanding viral adaptation, bacterial resistance, and host pathogen interactions. In addition, comparative genomics contributes to the broader framework of personalized medicine. By understanding genetic variation in evolutionary and functional context, researchers can better interpret which variants are likely important for health and disease. If you look at the slide, the key message is that evolutionary analysis is not separate from medicine. It directly informs it. To conclude, let us bring these ideas together. Comparative genomics connects many types of biological data into one coherent framework. Genome browsers help us visualize conservation and sequence organization. Orthology databases help us identify evolutionary relationships. Functional annotation resources help us interpret biological roles, and together these tools reveal which parts of life are deeply conserved and which parts have changed over time. So comparative genomics is not just about comparing species. It is about understanding how shared ancestry, functional conservation, and evolutionary change shape biology. If you look at the final slide, the complete workflow moves from genomic comparison to biological meaning. That is the real power of comparative genomics. Thank you.